Drug discovery begins with an idea, or when there is a human disease without appropriate treatment available. The process from the initial idea to when the drug reaches the market is a long and expensive one. The entire development process takes between 12 to 15 years, involves screening of over 5,000 compounds, requires testing on thousands of human subjects, and costs around $2 billion. Sounds like a lot, but top-selling drugs make around $8 billion in sales in 1 year alone. So not only do these new drugs provide a better quality of life for patients with diseases, they are also huge money makers for pharmaceutical companies. The process can be divided up into 5 main stages: discovery, preclinical studies, clinical trials, regulatory submission, and post-market surveillance. The entire development process of a new drug is highly regulated by agencies like the FDA, TGA, EMA, and ICH. All these regulatory authorities ensure that the new drug that enters the market is safe, effective, and of high quality. They do this through laws published in the Code of Federal Regulations Title 21, as well as ICH guidelines. The first stage of drug discovery is identifying the target, the molecular structure in the body that the new drug will potentially act at to produce an effect. Most often these are receptors, for example, G protein-coupled receptors. The next step is to find lead compounds that will interact and show desired activity at our target site. High-throughput screening, or HTS, is a fast screening technique that can screen up to hundreds of thousands of compounds a day. This system works by having samples of compounds and reagents deposited into tiny wells on a plate, which are then incubated. A range of detectors like fluorescence and luminescence can then be used to observe hit. These hit compounds can then be further optimized with the possibility of becoming a new drug candidate. Most drugs that reach the market have a recommended shelf life of 3 years. In order to guarantee shelf life for that long, lead compounds must undergo stability testing at different temperatures. Drug companies can also perform accelerated stability studies by storing drugs at elevated temperatures beyond what they are likely to experience once they are on the market. During drug discovery, sponsors can file for a patent to IP Australia or the Patent and Trademark Office in the US on Form 3542. This enables them to protect their invention of their new drug, as well as preventing others from using or making their drug until the patent expires. This is typically 20 years or sometimes up to 25 years. Before preclinical testing, pharmaceutical companies can construct a mathematical model that predicts in vivo bioavailability of the drug from in vitro tests. This is called IVIVC. This correlation is advantageous as it can save drug companies money by predicting if a drug is likely to fail during earlier stages of the drug development process. Generic drug companies who come in when the innovative drug reaches the market can also use IVIVC data to support their application for biowaivers. Preclinical studies involve testing lead compounds in vitro and in vivo in animals. These studies help determine the toxicity of the drug and provide a good idea of starting doses for clinical trials. This stage can take between 3 to 5 years and involves testing of less than 250 compounds to further try and narrow down the search to one final drug candidate. Regulatory authorities provide laws on good laboratory practice, or GLP, to ensure safety of animals during testing, as well as maximising collection of beneficial data. Towards the end of preclinical testing, an investigation new drug application is filed to the FDA in order to receive permission for human clinical testing. The application provides all applicable data collected on pharmacokinetics, and pharmacodynamics during preclinical testing. In Australia, an IND is submitted via a different format. Clinical trials can be granted under a notification scheme or an exemption scheme. Majority of trials are conducted under the exemption scheme as they are much cheaper. However, the sponsor does not receive any feedback or advice if their application is rejected. Before human clinical trials begin, it is important to finalise the dosage form of the drug, whether it is a tablet, capsule, inhaler or something else, as the route of delivery can have a great effect on the drug's bioavailability. Clinical trials are regulated through good clinical practice standards published by regulatory agencies to protect the rights and safety of human test subjects. Phase 0 clinical testing involves a small population of around 10 people to administer a microdose and ensure the dose determined from preclinical studies is safe in humans. Phase 1 is used to determine drug safety by evaluating its pharmacokinetic profile. Doses may be increased until side effects are observed in order to determine the highest tolerated dose. This phase involves 20 to 100 healthy human subjects, apart from cancer drugs, which are studied on a population of cancer patients who don't respond to standard treatment. Phase 2 involves testing the drug's safety and efficacy and is conducted in up to 1,000 people who have the target disease. This is typically when the optimal dose of the drug is established. Phase 3 also studies drug safety and efficacy, however in a much larger population of human subjects with the disease, so that statistical data can be shown to regulatory authorities to prove the drug's safety. The data gathered from Phase 3 trials is also critical in terms of the claims the drug company can make about their product. Information regarding doses, directions of use, excipients and warnings are prepared for listing on the drug label. Upon completion of clinical trials, a new drug application, or NDA, is filed to the regulatory authorities for review so that your drug can be improved for release onto the market. For the regulatory authorities may respond with an approvable letter requiring additional testing or information. The NDA is submitted in conjunction with the Common Technical Document, an application dossier demonstrating the drug's quality, safety and efficacy. Before regulatory authorities approve your NDA application, they are likely to visit your testing and manufacturing facility for a pre-approval inspection. They ensure that all current good manufacturing practices are followed, from proper training of staff and personnel to quality assurance in testing and production. When your drug becomes approved, it is ready for manufacturing and market launch. In Australia, drugs are registered on the Australian Register of Therapeutic Goods. The ARTG can be searched by anyone from the public and provides summary information about the drug's dose, storage, directions of use, and more. However, drug testing and control does not stop when your drug reaches the market. Phase 4 trials are conducted for as long as the drug is being sold to the public. Drug companies are required to continually submit long-term safety data to regulatory authorities. If you do experience any adverse events after taking your new drug, it is important that you notify your country's regulatory agency, as it can provide crucial data in terms of the drug's safety or interactions with other pharmaceutical compounds that were not able to be observed in clinical trials. If it is identified that a drug no longer meets safety or quality standards, it will undergo a review and may eventually be recalled or withdrawn from the market. A recall of a drug is very difficult and complicated, and that is why regulatory agencies have strict laws and guidelines during the drug's development process. Once an innovator's drug patent expires, generic drug companies are able to manufacture your drug and will tend to sell it for a much lower price. This is because if a generic drug company can prove that a drug is bioequivalent to the innovator drug, they can omit certain areas of testing. to prove their drug is bioequivalent, it must show the same rate and extent of absorption. Therefore, graphs showing concentration of drug in plasma first time are able to correlate whether the drugs are bioequivalent or not.